Hello everyone, today let us learn about polymerase chain reaction in short. It is a technique used for generating large quantities of specified DNA. It includes three steps. They are denaturation, annealing with primers, amplification. Principle: it is a double stranded DNA, so it is denatured to separate into two strands. Each single strand is allowed to hybridize with the primer. Using this, DNA is synthesized. These steps repeated again and again to generate multiple forms of target DNA. Requirements of PCR: we need an enzyme DNA polymerase, TAC polymerase, that is Thermus aquaticus, target DNA, 2 primers, all the 4 nucleotide bases. So these are the steps of polymerase chain reactions. So here we have a double stranded DNA, all the requirements, that is specified target double stranded DNA. We have a primer, we have a DNTPs, all the 4 nucleotide bases, and the TAC polymerase. Here denaturation is done and separate the double stranded DNA are separated to get a 2 single strands. It is followed by annealing, that is addition of primers at the temperature 50 degrees Celsius. So this is the first cycle, and this is followed by extension, where addition of DNTPs and growth of primer complementary to the sequences present in the parent DNA strand. And in this step we use an enzyme called TAC polymerase enzyme, which is stable at the temperature 72 degrees Celsius. So this occurs at the temperature 72 degrees Celsius. So with this we get a 2 DNA from this single DNA we get 2 DNAs, double stranded DNA. From this we get 2 double stranded DNA, and from this DNA again the cycle is repeated, like denaturation, annealing, and extensions. So from this DNA we get 2 double stranded DNA, from this we get 2, like this the cycle gets repeated to get millions and millions of specified DNA. So at the end of one cycle we get 4 strands. At the end of 2 cycles we get 8 strands. At the end of 20 cycles 1 million strands. So it is sensitive and specific technique. So what are the types of polymerase chain reaction? Reverse transcriptase, PCR, nested PCR, real-time PCR, multiplex PCR. Applications: prenatal diagnosis of inherited disorders like sickle cell anemia, beta thalassemia, phenylketonuria. It helps for the diagnosis of retroviral infections like HIV, diagnosis of bacterial infections like tuberculosis, diagnosis of cancer, DNA sequencing, study of evolutions. It is used in forensic medicines. Thank you.